Okay, so good morning, everybody. And I hope that by now you have all watched those videos on YouTube on how to solve Hardy-Weinberg equilibrium problems, and that you have made a start on the problem set that I gave you. Remember, submit that problem set to me by Monday or Tuesday is fine, at the end of the semester. And that counts for 2 quiz grades, okay? And those are not particularly difficult problems. I'll give you, and I will post the answer sheet on Tuesday so that you can use them to study. Okay, so we're having a look at the Hardy-Weinberg equilibrium equations and what they describe. Hardy-Weinberg equilibrium applies only under very strict theoretical conditions, and we saw the conditions were no immigration, no emigration, no natural selection, no mutation, etc., etc., etc. All of those are actually unrealistic expectations. Hardy-Weinberg equilibrium, this is a theoretical construct. We can apply it in certain circumstances, as you have seen from the problem set. But what Hardy-Weinberg equilibrium implies is that from generation to generation, the proportion of alleles remains unchanged. And that is obviously untrue. If that was true, no evolution would ever take place. But in fact, we realize that there are 3 situations where Hardy-Weinberg equilibrium is disturbed. And this is a continual drive throughout all of biology, because the one major way in which Hardy-Weinberg equilibrium would be disturbed, the one major way in which allele frequencies are altered in populations is by natural selection. So all of our discussion about natural selection, Darwin's ideas and everything else, we can rethink all of those in terms of changing allele frequencies in a population. Okay, natural selection is the one major way in which Hardy-Weinberg equilibrium becomes disturbed. There are 2 other circumstances where it will be disturbed as well. The first is called genetic drift. We'll define and talk about it later. And the third is gene flow. That is when we have, for example, we may have 2 different populations separate from one another. Each of them proceeds along their own evolutionary path. They develop their own sets of alleles. They may well establish Hardy-Weinberg equilibrium for those populations. But then if we join the 2 populations together, we get a mixing of new alleles, and any disturbance of allele frequency is evolution, and any change in those allele frequencies, Hardy-Weinberg equilibrium has been disturbed. So gene flow is the third way in which we recognize evolution's taken place because of changes in allele frequency. So let's have a look at them in more detail. The first, natural selection, we've talked about at length in terms of observation, of Darwin's observations, etc., etc. But let's rethink it a little bit in terms of allele frequency and of what we have heard about Hardy-Weinberg equilibrium. The natural selection works by selecting 4 particular alleles. In other words, natural selection takes organisms which have advantageous alleles, for example, and it selects for those advantageous alleles because they allow the organism to suit better in its environment. Because it suits better in its environment, that organism will reproduce more. Because those organisms reproduce more, their alleles become more and more common in the population. The disadvantageous alleles may still be present in the population. They may be present, for example, as a recessive, But natural selection favors advantageous alleles. And so it moves the allele frequencies. It increases the allele frequencies of those which are favorable. And that we refer to as adaptive evolution. Natural selection continually improves the match between an organism and its environment. So that improvement in match occurs because those organisms have the advantageous allele. So there are many examples which we actually can see in action, which illustrate this. As for example, in use of insecticides, the conference of resistance to insecticides in insects. That is an example of natural selection in operation. An allele that confers resistance to an insecticide like DDT. We can actually go to a population after a time that the population has been exposed to DDT or any other insecticide, and actually look for the alleles which confer resistance and see that they become more and more and more common in the population. It's a huge problem for us because that is natural selection, which actually operates quite rapidly because there is a short generation time. Similar idea for resistance in bacteria. Resistance in bacteria can be spread through a whole bacterial population very quickly because those resistance alleles are selected for. So, sorry, I just need to, there we are. So that kind of evolution by natural selection, what is happening is that it involves chance, and it then requires a sorting mechanism. And natural selection is the sorting mechanism. The new genetic variations, the new alleles, may be extremely rare in the beginning because they arise by chance. They arise by mutation. They may be extremely rare. But if they're advantageous, they will be sorted because those organisms that possess them are at an advantage in the environment. And so the natural selection selects for those alleles by sorting out those individuals which have them. This is how natural selection operates on an individual, but evolution, the change in proportion of alleles in the population, operates at the population level. Natural selection is the only one of the three mechanisms that we're going to talk about, which consistently increases the frequency of alleles, which are going to provide a reproductive advantage. In other words, only natural selection increases fitness. Fitness being the ability of the organism to reproduce. Those which are more fit reproduce more. And only natural selection moves the population in that direction. The other two, genetic drift and gene flow, do not have that ability. As you'll see, they're more random processes, as we'll see. So let's have a look. These are just simple examples, and I think that there are more in your text for you to read about. But in natural selection, actually there are different aspects to natural selection that we recognize. Let's just take a simple example of rodents in a varied environment, let's say, and there is a range of different alleles which confer coat color. And as in any healthy population, there is variability in the population, but the frequency of alleles is here, and it favors these which are, well, just call them the median animals. Okay, but within the population, there are those, for example, which are much darker. There are those which are much lighter. Now, this population will remain like this as long as their environment remains stable. But environments seldom do remain stable, especially over a long time. Changes can take place in the environment. For one reason or another, new predator comes in, climatic change. All of these things might alter an environment that a population is living in. And you would see new selection pressures being exerted on the population. Our population here, old population, and it's got variability of coat color. Now something comes along to disturb the equilibrium. If we were mapping this with Hardy-Weinberg, we would just assume that this went from generation to generation with no change until some environmental change takes place. And now there are three ways in which we could envision this population being disturbed. The first is that these, on one end, the alleles here are selected against. Let's say that the environment changes and the ground becomes darker for some reason. Okay, these are the lighter are going to be visible to predators and might be taken out more easily. In that this is the most common because this is called directional selection. And what is happening is one set of alleles is being strongly selected against. One set of alleles is being strongly selected for. We said that our environment has changed, so the ground has become darker for some reason or another. These dark animals have an advantage because they are not visible to predators. That is a process of evolution. The proportion of alleles is changing. The proportion of alleles for dark fur are being selected for, and this we call directional selection. This original population, when we see the frequency of the alleles, we'll see darker alleles becoming more and more frequent. We can also envision a situation where, in fact, these ones in the middle here are selected against, where those at the extremes are selected for. This we refer to as disruptive selection. And now, disruptive selection is very, very important. Disruptive selection often, in real life, can envision it happening. For example, if a population becomes split into two geographic areas, maybe some separation takes place here, where these get separated off to white sand, say, while these ones get separated off to dark sands or to rocks or something like that, where the ones in the middle are selected against, the ones at the extremes are selected for. And this can be a very important mechanism in speciation, because if this disruptive selection persists and these alleles are lost, we may in fact increase the proportion of these alleles and these alleles to such an extent that these populations separate entirely from one another and become different species over geologic time. The third type of selection is where the extremes, the very light and the very dark, are selected against. And this we call stabilizing selection. And in stabilizing selection, in fact, the number of alleles in the population begins to decrease. The population becomes more and more homogeneous, and as these alleles are lost from the population, so the population becomes more and more homogeneous. This we refer to stabilizing selection. Okay, so there's the three: directional, disruptive, and stabilizing selection. But they're all natural selection processes. So just to repeat, what we've said it frequently, that natural selection is going to increase the frequency of alleles, which enhance survival and reproduction. Natural selection operates by increasing fitness. It increases the reproductive capability of organisms that have a favorable allele. And natural selection is directional in that it continually improves the ability of organisms to survive in their environment. This adaptive process is continual. It's a continual process because there are very, very few environments which are absolutely stable over geologic time. They may be stable over our lifetime, so we might not see processes taking place, but over the course of millennia, environments may change very radically. And this drives continual adaptation of organisms in those environments. So natural selection drives evolution continually. Now, the other two processes that we're going to talk about, genetic drift and gene flow, do not lead to adaptive evolution. They are far more random processes because they can actually increase or decrease a match between an organism and its environment. They can actually end up removing individuals which were better, which were fitter for an environment. And that may sound strange, but you'll see exactly what I mean. Genetic drift is a process whereby alleles fluctuate unpredictably. They fluctuate at random because random processes affect them. You have a population of flowers, let's say, and just as all the flowers are getting ready to reproduce, they're busy setting seed, a rock falls right in the middle of the population and smashes 2/3 of the population and takes out alleles which they carried and leaves other flowers at random with their alleles. You may have changed the frequency of alleles simply by chance, by accident. That would be an example of genetic drift. And genetic drift may in fact take out organisms purely by chance. It takes out organisms which are fitter, and that their genes are lost to the population because of it. There are a couple of circumstances that we'll see where we believe genetic drift has actually operated on real populations. Genetic drift tends to reduce genetic variation. It tends to remove alleles. We can't really envision genetic drift process whereby alleles would be increased. The other important thing about genetic drift is it has its strongest impact on small populations. If you have a very large population and a wide distribution of alleles, taking out a few members of the population is not going to affect the overall proportion of alleles. It's when a population is small that taking out individuals by chance actually has a greater impact on the distribution of alleles in that population. And let's just take this example here. Here are our red and white and pink flowers again, our CR and CW. And here's our population. It's not a very big population, but if you count them up, there are 20 alleles altogether, and there are 14 CRs, and there are 6 CWs in that population. Now, if this stays in Hardy-Weinberg equilibrium, then on our next generation, what are going to be the proportions are going to be exactly the same. The frequency of CR, 0.7, 14 over 20, 0.7. The frequency of Q of the CW is going to be 6 over 20, 0.3. Okay, and if this was in Hardy-Weinberg equilibrium, there's nothing happening, no immigration, no da 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 da. The next generation would be the same. However, purely by chance, maybe certain plants got eaten before they could reproduce by a predator, and only 5 plants leave offspring, and that's this one, this one, this one, this one, and this one. Only these plants get to contribute their genes, their alleles, to the next generation. And when we look and we go there and we count, we find they didn't obey Hardy-Weinberg because the proportion of alleles changed from one generation to the next. There's no selection pressure here. It's simply that by chance, these ones here got eaten. Or whatever, for whatever reason, by pure chance, they didn't reproduce that year. And they didn't get to hand on their alleles to the next generation. And the proportion of alleles changed. If a proportion of alleles changed, evolution is taking place. But in this case, it's taking place not because of a selective pressure, but because by pure chance, genetic drift occurred. Now, this is a small population. Now, let's say only 2 plants actually reproduce this next year, this one and this one. Well, look at what they are. They're homozygotes. They're homozygous CR. If only those 2 reproduce the next year, your next generation, that CR gene we say now is fixed. The entire population consists purely of these, of CR. And over a space of 3 generations, our proportion of alleles has changed. Look, the frequency of CR, 0.7, 0.5 to 1. It's the entire population. Frequency of CW, 0.3, 0.5, so it's half there, to 0 in the next generation. So this could be a very, very important means of evolution. It's actually eliminating an allele entirely from a population, but it leaves this population monotypic. And that is a very dangerous thing, because what if, for instance, a disease comes in and the disease specifically attacks plants with the CR allele? Well, if that was to happen, the entire population would be lost. In fact, populations with no variability are vulnerable populations. They are vulnerable to all sorts of environmental pressures because they don't have the alleles present. For this, what needs to happen to this population? For it to acquire variability, with time, mutation will begin to add in alleles again. And then those get sorted. Then this population is again available for natural selection, not for genetic drift in this case, but for natural selection. This is an example of genetic drift. And it's a real situation. We see some specific instances of this. The first is called the founder effect. And it's when in moving from one generation to another, just a few members of a population are actually selected. And it's purely by chance, right? And then a population gets established just from those few original organisms. And all that's available for a long time until mutation begins to add in new alleles, all that is available for a long time are the alleles which those founder organisms provided to the population. And we see this kind of effect. For example, this is how the Galapagos got populated with finches, right? The Galapagos got populated with just a few individuals who brought in just a few alleles. what happens with founder populations very often is that the intraspecific competition then becomes so intense. What's happening is you've got many individual organisms with very similar genetic makeup, and they are competing vigorously for resources. Now, natural selection begins to operate very rapidly. It begins to strongly favor even those which, with the smallest little advantage in the population, get favored. But your initial population, will all look very similar to one another. The situation where we have a founder population and then very rapid natural selection because of the intensity of competition, we refer to as adaptive radiation. And it's very typical of situations like those in the Galapagos. That is how a founder population of finches on the Galapagos, all initially very, very similar to one another. The population got too large for the environment to support. The finches began to compete with one another, and those with slight advantages began to occupy slightly different ecological niches and succeed there. And they acquired characters which differed from the rest of the population. And very rapidly, on an island situation like that, we see speciation, we see divergence into many different groups. We see very rapid natural selection operating, but it operates from the founder effect at the very beginning. And there is another instance of genetic drift that we do know about, and this is called the bottleneck effect, where there is a sudden selection, a sudden reduction rather, in population size. It can be for many different reasons, but very often due to geographic change or emigration or something like that. You just get a few. It's in a way similar to the founder effect, and often we believe a major portion of the population is actually lost, leaving only a few individuals from the original population. And those, obviously, if there's only a few individuals from a formerly very large population, they only have a restricted number of alleles. They then breed, and what we observe very often is that they, if they do not undergo adaptive radiation, as they would in an island situation, something like that, we often observe that their genetics are very, very similar. And we know of several populations where this has occurred. So here's the graphic description of it. Here in this, in this original population here, we've got a whole mixture of alleles. Okay, the blue and the white appear to be more common alleles, but there are other alleles present as well. So this population has a lot of variability in it, as one would expect. Some event occurs. This is genetic drift, so it's basically at random, but some event occurs which selects just a few individuals from that population. And this is a bottlenecking event. When we look at the surviving population, we had a very large population before with lots of variability. We are left with a very small population with only a few alleles. And the proportion of alleles is completely different to what it was in the whole population. This population now goes on to reproduce and everything else, but all that they have to work with is a restricted number of alleles in the population. There's very little variability. Most of the population are genetically extremely similar. And we know of instances where this has occurred. Some of them fairly recent, others more ancient. We believe, for example, that all the world's cheetah populations have arisen from a very, very few individuals who survived some cataclysmic event they evolved in Africa and Asia, but there was some cataclysmic event which very severely restricted the population of cheetahs. It may have been disease or something which is more. There's a lot of speculation about what occurred, but what we do know is all cheetahs that currently have extremely similar genetic makeup. They have all arisen from a very small population of surviving cheetahs that survived some event, but in an ancient, predating human. It's not predation by humans or anything like this. Here's another instance, and this is the elephant seals. Elephant seals, this is largely due to human impact. Elephant seals were just about rendered extinct by hunting, by humans, but then came under protection, under worldwide protection, when it became apparent that the populations were close to extinction. So all the elephant seals that we see now, and the populations are getting quite large, all of them have arisen from those few survivors. So it's like a founder effect, excepting that this is some specific event which selected just a few who survived the human predation to found new populations. They're all very, very similar. But the last population may interest you because there's a strong suggestion that human beings ourselves underwent a bottleneck event. And so, for example, if we look at human populations worldwide, there's actually genetically, we are all extremely similar to one another. The species Homo sapiens, all of the individuals are extremely genetically similar. And with many scientists, geneticists will suggest that we underwent a bottleneck event. And exactly what it was is not clear, but it may well be that it was our exit from Africa, that the species Homo sapiens evolved in Africa, and that a very few of them managed to leave Africa and establish populations in the rest of the world, and that event itself was a bottleneck event, which has led to us all being very, very genetically similar to one another. Just interesting to speculate about. So in summary, for genetic drift's really significant only to small populations. And genetic drift causes allele frequencies to change, but at random. There's no directionality to it, the way there is for natural selection. And it leads to a loss of genetic variability in populations as individuals are taken out at random. There's one unfortunate effect of genetic drift, and that is because it takes alleles out at random. It can take out good alleles, and it can leave behind deleterious alleles, and it can increase the frequency of those deleterious alleles in a population. It can render a population vulnerable. As I said before, it can render that population red flowers. It could render a population very vulnerable to something like disease, things like that, because they've lost advantageous alleles within the population. Okay, so the last way in which we can observe evolution occurring through change of allele frequencies is if we have gene flow. And that is movement among populations. So what we're doing is we're moving alleles into and out of populations, either by emigration or emigration. And this, in actual fact, this can occur conceivably in many different populations which are slightly separated from one another, let's say. we had two populations which are separated from one another. They proceed along their own evolutionary paths and their own environment, under their own environmental conditions. But if we then allow them to mingle, then we change the proportion of alleles in the whole population. But there's another instance where this can conceivably occur. I mean, not with the individuals themselves mixing, but with gametes mixing. Just think, for example, about pollen. Pollen is a gamete. Pollen is one of the gametes, is male gametes, contains male gametes of plants, and is blown widely through with the wind. So, for example, you can conceive of a population with a set of advantageous alleles, contributing those alleles to another population because pollen from them was blown in to fertilize those plants. So gene flow, as strangely as it may seem, it actually reduces variation in populations. Why? Because it blends populations. It blends populations together. And once those populations are blended like that, then natural selection may continue to operate to favor select certain alleles. So again, we have gene flow, we have evolution purely by gene flow, but then we set up a population where natural selection again can begin to operate. Overall, we do believe that gene flow does increase fitness of the whole population because it creates a population with wider variability, but then natural selection has more alleles to operate on. And so we can see examples of this. You may know, for example, that there is a tremendous amount of concern about mosquito species spreading through the United States, for example, which can carry particularly viral diseases, but also diseases like malaria, those sorts of things. So this is actually something which is observed. You may, for example, have populations of mosquitoes in one area, which are being treated with insecticides. Those insecticides provide a very strong selection pressure for insects which are resistant to the insecticide. That develops in a particular region. But if those mosquitoes spread into regions where no insecticide has been used, they bring their alleles with them. The gene flow actually contributes alleles to the population, even if even if that population has not yet been exposed to insecticide. Now, if we go and use insecticide there, we find that a higher proportion of the population are resistant to insecticide than we would expect because there has been gene flow into the population. We also believe that gene flow is very, very important in human evolution. It should be clear to you that first of all, we talked about we may have had a bottleneck effect. Originally, we all relatively genetically similar, but variation has occurred. And if we look regionally at ethnic variability, I'll give you an example that, for example, in our immune systems, there is variability which is strictly dependent on where human beings have resided for a very long time. So there is genetic variability in the immune systems. If we now allow gene flow to take place, those advantageous immunological genes can be contributed to other human populations. So gene flow in human populations has been extremely advantageous over our history. I don't know what this is. There we go. Okay, so let's now leave that aside for a moment, and I just want to talk a little bit about some special instances of natural selection, which have their own interest. The first is this question in natural selection of sexual selection. Sexual selection is selection by a mate. Now, you remember something. When we talked about Hardy-Weinberg equilibrium, we said there was, in Hardy-Weinberg equilibrium populations, there's no sexual selection, right? Any one gender can mate with the opposite gender, irrespective of the individuals. But that is not the way it operates in many organisms, especially animals. And instead, there is a selection for likelihood of mating success. So, and that is what sexual selection is. It's a mating. It is a mating selection of a mate for the likelihood that they are more successful than others. And the first thing that this does is it results in sexual dimorphism, in marked differences between the sexes. And the dimorphism is often an amplification of some morphological feature which implies reproductive success. So, for example, if we look at the peacock, how did the peacock male end up getting this huge tail? Well, what drove it was the female. The female is actually selecting the male. The male may puff himself up and flash a tail and think that he's doing the selection, but in actual fact, it's the female doing the selection. And she originally selected maybe male with a small tail like this, but that male with the tail that was attracted to the female, he had good strong sperm, or maybe he was strong himself and could protect her, maybe whatever. Originally had some reproductive advantage. He was more fit. What did his offspring look like? Well, his offspring are going to carry his genes. Our original male, maybe with a tail this big, that tail becomes a marker of reproductive success. What do females begin to select for? They're beginning to select for that marker. They begin to select for marker. And the advantage given by bigger tails is increased and increased and increased until the male has this immense tail. It may, in fact, be so great that his individual suitedness for his environment is impacted. Here, this may be disadvantageous to the male, but it is selected for because it marks him as being very fit, as being an organism who will produce lots of offspring. And the selection is done by the female. This drives two things. It drives, first of all, sexual dimorphism, marks out the one sex from another very distinctly. But it also can produce these elaborate, exaggerated symbols, if you like, of the likelihood that he will be a successful mate. So that's how, in fact, this huge tail is disadvantageous to the individual. It makes flight difficult, and everything, and probably makes evading predators difficult. But it gets him lots and lots of babies. It gets him to mate with many different females, and he produces many offspring. And his male offspring all inherit his big tail. So this can reach. This doesn't only have to be morphological, by the way. It can also be kind of behavioral. This is a satin bowerbird. The bowerbirds, there are several species, but they build these elaborate, not nests. They are advertising stations. They're actually called a lek, but it's a place where he will lure a female to come in and examine, and she judges in a way his likelihood of his reproductive success by the nature of the display that he lays down for her here. And this is his ability to do so is genetically determined. Those who do it better, well, they get more mates. And their progeny inherit the ability to do this even better and better and better and better, and so on and so on, until we end up with this incredible, elaborate system. It has this is not producing babies. This is not sheltering babies. This is not doing anything except advertising. And it's advertising his ability to produce young. And so those who do better displays, they are selected for, and those genes are handed on to their progeny. It's a form of selection, but it is selection through bisexual selection. Here's another one. This is extraordinary. These are stalk-eyed flies, and in stalk-eyed flies, the males have these incredible stalks on the eyes, and they do a dance for the females to attract females, and those with longer stalks on the eyes, they attract more females. And because they attract more females, the genes for long stalk get handed on, and this has become more and more and more exaggerated. If you actually look at populations of stalk-eyed flies, you find that this actually there's a limit, and there's a point at which it becomes so disadvantageous to the male that too exaggerated an eye stalk actually becomes disadvantageous, and that male will be lost from the population. So there is some sort of balancing selection here as well that is going on, selecting for these extraordinary long eye stalks which have no function other than to attract a female. Okay, so the last thing I want to talk about is you'll have realized when we did Hardy-Weinberg equilibrium that we're in any population like that, we always have a certain proportion of our population are likely to be heterozygous at any locus. And heterozygosity is actually very common for many, many, many of our genes. And it turns out that, well, if you think about it this way, why doesn't a gene, an allele, just disappear from a population? If one allele is more and confers some advantage, why doesn't the other allele simply disappear from the population? Why isn't it selected against completely? It turns out that in fact very often the heterozygote is at an advantage over homozygotes. And for that reason, natural selection tends to preserve the diversity. It will tend to maintain different alleles at the same locus, because being a heterozygote is more advantageous than being a homozygote. And we have a very good example of this, and that is in the mutation which causes the disease of sickle cell anemia. And I think I've mentioned this before, but I'll just go through it again. So let's just go here and have a look. The mutation that causes sickle cell anemia replaces an adenine for a thymine in the gene, and it changes the amino acid that is coded for here from glutamine to valine. This is the gene which is going to produce hemoglobin. Now, hemoglobin picks up oxygen, transports it around the body, and it drops oxygen in areas of the body which are low in oxygen. Normal hemoglobin is flexible, and the entire red blood corpuscle that carries it is flexible and can make its way through the very fine capillaries quite easily. And that is where the oxygen tends to be dropped more. In the mutant hemoglobin, when it reaches low oxygen tensions in the capillaries, for example, it actually begins to form a fiber, a more rigid fiber, which does not change shape easily and does not, it's a permanent change. It doesn't return to the shape when it gets to rich oxygen. It's a permanent change in the red blood corpuscle, and it causes the red blood corpuscle to cave in and collapse and become more rigid. And they can't make their way through the capillaries. These can deform themselves, can be deformed to make their way easily through the capillaries. These can't, they are rigid, and they begin to pack the capillaries. And once they pack the capillaries like this, they set up an inflammatory condition in the capillaries. Remember, these capillaries may be in the periphery, maybe in your fingers, toes, etc., but they're also present in all of your organs, throughout your body, etc. So there are many regions of the body where in sickle cell anemia, these inflammatory conditions are set up where the blood begins to clot within the capillaries like this. And it can be extremely dangerous. There is a single site, there's a single locus at which this change can take place. And so this sickle cell allele is present in the population, and it is present together with the normal hemoglobin allele population. And if you look at the frequency of the sickle cell allele, you see that it's up to 15% of the population. But just think about this. If I can get back, sorry. Just think about this. If you are homozygous for this allele, all of your hemoglobin is sickle hemoglobin. And it's extremely dangerous. It's extremely dangerous. It would seem that natural selection would operate very, very strongly against that. And indeed, there is strong selection pressure against individuals who are homozygous. They tend to be prone to severe illness. They don't reproduce probably well. Many of people who are homozygous for the sickle cell may die before they become reproductive. So why isn't that allele simply removed from the population? Well, the answer is that if you are heterozygous, if you have one normal hemoglobin gene and one sickle cell allele, you are actually at an advantage. And the reason is that the selection pressures are arising in areas where malaria is present. And the malaria parasite cannot infect sickle cells. It cannot infect red blood corpuscles, which have a sickle hemoglobin. So first of all, people who are homozygous, they're at an advantage because they're not going to get infected by malaria. But they're at a huge disadvantage because they are homozygous for the sickle cell hemoglobin. But people who are heterozygous are at an advantage. First of all, they have a reduced susceptibility to malaria because some of their red blood corpuscles have sickle hemoglobin. And they're at an advantage because they have a normal hemoglobin gene as well, which gives them enough normal hemoglobin for them to be able to function. So the heterozygotes are at an advantage over homozygote normal. Homozygote normal are at a disadvantage because they're much more susceptible to malaria. So what we observe in actual fact, the sickle cell allele is more common in areas like this in West Africa, for example, to a certain extent in India as well, and some in the Arabian Peninsula, all areas high in one species of malaria called Plasmodium falciparum malaria. There's a strong association between survival of this allele, the sickle allele, in those areas where malaria is common, because the heterozygotes, people who have one normal and one sickle cell gene, are at an advantage because they have a lower infectivity by malaria. So that's the classic example. Okay, and that completes that for today. And I will see you then again on Monday.